Alright. Hello everyone. Let's see, there are significantly fewer people logged in today. I'll take that to mean people are probably studying, so that's good. One way or another, I will be posting these on YouTube like last time. So you can go over there and take a look if there's anything you'd like to see again. Okay. Got a few emails that I got earlier in today, and I'm just going to go through those, and if I get any more emails I will answer them, similar to yesterday. And if we go through a period of half an hour with no questions, we will sign off, and I will answer all the rest of your questions just through email. Alright, so let us begin. Alright, question 1. Hi, can you explain to me, I'm having trouble understanding this, how might an agonist have an inhibitory action on a neuron? So that is a good question. So, the way an agonist might have an inhibitory action on a neuron is, say that you had something. So remember, there are excitatory receptors, and those are glutamatergic, and then there are inhibitory receptors, and those are GABAergic. So you have glutamate, which is excitatory, and you have GABA, which is inhibitory. So when you have an activation of either of these receptors, you're either going to get excitatory action or you're going to get inhibitory action on the inside of the neuron. So say, for instance, you had an agonist that was binding to a glutamatergic receptor. Well, you would have excitatory action. You would have an excitation of the neuronal membrane and an increase in charge. However, say that you had an agonist that bound to a GABA receptor. In that case, you would have an opening of the GABA receptor and a net inhibitory action on the neuron. So, the way an agonist might have an inhibitory action in a neuron is if that agonist activated an inhibitory receptor. You had a GABA receptor, just draw it here, and you have a GABA agonist, say ethanol, comes in and binds to it. Then you can have something like this going into the cell. So chloride would have, because it is an anion, it would flow into the cell, and it would make it more difficult to fire an action potential by decreasing the voltage. So that's how an agonist can have an inhibitory action. Alright. Next question. Alright. The presynaptic cleft is on the axon terminal on cell A, and the neurotransmitters spill into the synaptic cleft onto receptors of the postsynaptic membrane of cell B. Is this correct? Yes, except you used one word. You used presynaptic cleft. What actually would be is presynaptic terminal. So just to illustrate this, you would have, oh goodness, here you'd have a neuron. This would be, as you called it, cell A, and this would be the presynaptic terminal. And so what you would have is all these vesicles here, on the presynaptic terminal. And here, as you correctly described it, would be cell B with the postsynaptic membrane. And then this area here is the synaptic cleft. So, if there's an activation of cell A, neurotransmitters will spill into the synaptic cleft over into cell B, which has the postsynaptic membrane, and these neurotransmitters will presumably bind to receptors and cause changes in the voltage of the postsynaptic membrane, changes in the voltage of cell B. So you are correct. The only thing is, instead of presynaptic cleft, it's a presynaptic terminal. Alright, moving right along. Alright, another question. What is the difference between the basal ganglia and the basal forebrain? For this exam, you don't need to know the difference. The basal forebrain consists of a much larger brain system. Namely, there's something called the basal forebrain cholinergic system, which provides cholinergic innervation to areas like the hippocampus and the prefrontal cortex, and actually the entire neocortex besides. But you don't have to worry about that. I know in the book it marks that as the basal forebrain. The globus pallidus, the putamen, and the caudate nucleus is the basal ganglia. That's what I would like you all to be familiar with, and that is involved in the initiation of voluntary motor movements. So do not worry about the basal forebrain. Alright, just got an email here. Hi, which kind of form of the words would you give in the exam? For example, gamma-aminobutyric acid or GABA? Definitely GABA. You will not you will not need to know the full name of GABA. Most of the terms in the exam are acronyms, if that makes it simpler. I will give you a fair warning, though, that you should pay attention to the acronyms on the exam, because sometimes there are going to be a few that may be very similar to one another, but they're just slightly different, and one might be right and the other one might be wrong. So if I don't think you're going to run into any problems where you don't know the entire, amino acid name of something, and you don't have an acronym, so don't worry about that. Alright. Next question is, could you go over the AMPA and NMDA receptors? Yes. Alright, let me just take a drink here. Okay. So, there are two kinds of neurotransmitters that will receive glutamate. Well, there are two kinds of receptors that will receive glutamate. So I'm just going to draw a cell here. We have an AMPA receptor. Again, you won't actually have to know what AMPA stands for. And then we have an NMDA receptor. I drew the NMDA receptor a little bit bigger because it is a little bit more interesting, and I wanted to convey that. Alright, so what happens when... Well, so what's the difference between these two just at rest? Well, specifically, in the NMDA receptor, there is a magnesium block at rest. So, just in any old day, if you had a neurotransmitter that would bind to the NMDA receptor, it's not going to open up. The gating for this receptor is restricted by the magnesium block. That's not the case for AMPA receptors. So if you have a neurotransmitter, like glutamate, an excitatory neurotransmitter, that comes in here, and then it binds to an AMPA receptor, I'm just going to say this is glutamate, binds to a receptor. That'll open the receptor up, and it'll allow for the flow of ions in and out. Say sodium in, and let's just say potassium is going to be going out. And so that's all good. And when an AMPA receptor opens up, and you have the flow of sodium into the cell, and keep in mind that since these AMPA receptors aren't voltage-gated sodium channels, it's not going to be such a dramatic increase. It's not going to be like there's a huge spike as there is with an action potential. It is actually what we would call graded potentials. It's just like the way the cell membrane is, just sits here, it goes up, and it goes down, it goes up, and it goes down based on the different excitatory and inhibitory input it's getting. So when it'd be going up would be when you have an activation of AMPA receptors, and there's an influx of sodium that'll change it, make it a little bit, it'll excite a little bit, then it'll go down a little bit, say when some chloride flows in, or if there's a period of inactivity, and so on and so forth. You don't have to worry about that for the exam, but that's just for your information. So when you have a neurotransmitter that binds to an AMPA receptor, like so, you're going to get these the free flow of ions through the open receptor. And what that's going to result in is it's going to result in an increase in the voltage of the neuron membrane. So that will result in an excitatory postsynaptic potential. So that would be an increase in the voltage. Now, what happens after the voltage increases by a certain amount? It'll hit a threshold, and at that point, this magnesium ion will dislodge. It'll go away. So AMPA receptors mediate these slight changes in membrane voltage up to a point. Eventually, the neuronal membrane will be depolarized to a certain extent, and that magnesium ion will pop out of the NMDA receptor. And actually, this illustration is so great because it's actually attached by a little bit of a string. It actually is on the bottom too, but it functions well for what we're talking about. And then once that happens, not only do you have the free flow of even more sodium and potassium, but you also have calcium flowing into the cell. Now, calcium, as we mentioned before, is very, 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 very important because it has a very low internal concentration just generally when the cell's at rest. So when calcium flows into the cell, that is a signal for all sorts of different things to happen. And we're not going to worry about all the different second messenger cascades that calcium affects and all the different proteins that it interacts with, but suffice it to say that in a similar way that when G-protein coupled receptors are activated, you get all these downstream effects that are huge, and they cause all of these changes that will shift around receptors and initiate these protein and enzyme changes that substantiate neurobiology of learning and memory and neuroplasticity. When calcium starts to flow into the cell, that's a signal for those things to happen as well. So the main difference between NMDA and AMPA receptors is NMDA receptors will allow for calcium to flow into the cell, and calcium initiates all sorts of changes that are necessary for neuroplasticity and, by extension, what we would consider learning and memory. And another difference is that at rest, NMDA receptors are, they do not have, there's no gating because they're blocked with a magnesium ion. Well, a magnesium particle. No, it's an ion. Yeah. And that magnesium ion has to be dislodged by the neuronal membrane reaching a certain voltage. So that's a very good question. Alright. I will. This is what I have for this particular topic on the study guide. Do you mind verifying it and answering question at the bottom? Okay, let me just give this a little read over real quick. Wow, you have a really great answer here. So this answer is correct. What you wrote, and what the student wrote and wanted me to confirm the correctness of, is as follows. In response to the question, describe the function of a G-protein coupled receptor and how this differs from an ion channel. Be sure to use the terms second messenger and modulation. The binding of a neurotransmitter to the receptor leads to the activation of G-proteins. Activated G-proteins activate effector proteins, which may be ion channels or enzymes that generate intracellular second messengers. That's correct. And I'll go ahead and illustrate that here just to explain it for those who just heard it. And the question that she asks is, can you explain modulation and how this is related? Well, all that modulation describes is how one of the qualities of G-protein coupled receptors in that they can interact with ion channels in the membrane, and they can modulate the activity. They can speed up the kinetics of the channel opening or closing. They can allow more ions to flow, or they can, maybe they could even turn the ion channel off. So modulation just simply describes how a G-protein coupled receptor can change and modulate the activity of other enzymes and other proteins, such as ion channels. And now I'm going to illustrate the statement you made, which was very correct, because I do not have another question right now. So in sort of working through this buffer period, since you gave me such a great explanation there, I will just illustrate it for the sake of the viewing audience, and then they will hopefully grasp it in the same way you do. So, again, I answered this yesterday, but describe the function of a G-protein coupled receptor and how this differs from an ion channel. Be sure to use the terms second messenger and modulation. So, an ion channel. We have a membrane here. We have an ion channel, and then we have a G-protein coupled receptor. So, what happens when a neurotransmitter binds to an ion channel? Ion channel opens up, free flow of ions in and out of the cell. Simple as that. Sometimes you get an ion channel that's particularly interesting. You'll have like a sodium channel, in which case there's no potassium that flows in and out, or you'll get a potassium channel, in which there's no sodium. and then you only have one ion flowing in or one ion flowing out. So, but that's really the extent of what ion channels do. They don't do much more than that. They just, neurotransmitter binds, ion channel opens, and you get these slight changes in voltage in the cell. G-protein coupled receptors are coupled to these little effector proteins down here. And when a neurotransmitter binds to them, I have a neurotransmitter coming in here. They will cause these effector proteins to go down and activate different proteins in the membrane. Those will activate other enzymes in the membrane, and those will cause things like ATP, will be turned into ADP. You'll have protein kinase A. It's going to phosphorylate onto itself and donate phosphate groups to different ion receptors. You don't need to know all this, but what happens, what I'm showing by illustrating all these things, is that when a G-protein coupled receptor is activated, it sends down these effector proteins, the G proteins, and they activate this, and they activate this, and they just go down, and they have these extreme effects throughout the cell. And we call these second steps here, we call them second messenger systems. So when you have an activation of intercellular second messengers, like it goes here, and then it activates, I think we call this a protein. Wow, it was a very poor attempt there. Just called protein. And then this activates an enzyme, and this activates another protein. So these second messenger cascades, where it goes down deeper, deeper, deeper into the cell, and it affects all these different processes that have these big changes that can be, as I said, it can be changing the function of ion receptors in the cell, or even adding certain things to other ion receptors and causing them to go into the membrane. So in effect, creating more receptors in the membrane from an intercellular storehouse. These are second messenger systems. So a neurotransmitter binds to a G-protein coupled receptor, activates effector proteins. They in turn activate enzymes, and they generate these intercellular second messenger cascades that cause this wide array of effects throughout the entire cell. And that's really all you need to know about that. Now, remember what I said with modulation. Say that one of these second messenger cascades activates an enzyme that goes up to this receptor, and now we've changed this receptor from a receptor that permits either both potassium and sodium to now it's just a receptor that only permits sodium. That would be a modulation of function. Now this is just a hypothetical. Don't look too deeply into it. But if if a G-protein coupled receptor activated an enzyme that caused this to switch from just a sodium and a potassium channel into strictly a sodium channel, that would be a modulation of its function. It's modulating the activity and the role of this particular ion channel. So that would be an example of modulation. Right, see that we got another question. Could you go over spatial and temporal synaptic integration? Yes. Alright, so remember we'll start with synaptic integration. So remember what synaptic integration is. You have, say in a cell, this is just, we're going to call this is a chart here, it's not actually a cell. Let's say, on the y-axis we have voltage, and we'll have the action potentials going here. So say that you had, well, I should be very careful, we're not talking about action potentials, we're talking about changes in the membrane potential. So these are graded changes. So you have cations flowing into the cell, you have an increase in positive charge. And what happens is you're going to have these, it's sitting at this, then it will slightly increase here, a positive charge increase, and then, there's more positive charge that's coming in, and then, you can see it gets even higher and higher and higher. And what's happening at each one of these stages is, say that you're having, positive charge here, positive charge here, positive charge here, positive charge. Then there's fewer positive charges here. Then you have even more positive charges that are building up here. And you have more. And then here you have a whole ton of positive charges that are causing this now very large depolarization. The integration of all of these positive charges here that causes this this big increase here, this graded potential, is synaptic integration. It is the integration of these different little positive increases in the membrane. Now, remember those positive increases are called excitatory postsynaptic potentials. So synaptic integration is very simply the adding together of multiple excitatory postsynaptic potentials to the effect that you have an increase in the graded potential here. So the question then becomes, how can these little depolarizations add up? So here we have a soma, and we have, we have our axon, and that's going to another cell, and we have our dendrites here, and then we have one dendrite here which we're going to be very interested in. So, say that you have positive charges that are coming in onto this dendrite. And they're going to be coming in, they're flowing in, and eventually they're going to propagate down into the soma, and then they're going to go down to the axon. They're going to terminate in those axon terminals. But right now we're focusing on the dendrite. So how do you get a very big signal? Remember, you're starting with these very small signals. You're starting with these very small little positive signals. But how do you turn that into a very big signal with a lot of area under that curve? How do you turn that into a big signal? Well, there's one of two ways. You can either do it by having a whole lot of signals occur in the exact same place. So positive, 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 positive. This will result in a very positive signal. And this is what is called temporal summation. So that when you have a high frequency of positive signals coming in here that result in a very large positive signal on the dendrite, that is what we will call temporal summation. Okay, and we call it temporal because they're all occurring close in time. So I believe in the book, it'll have this high frequency of positive signals will result in there being a very big positive signal here. right in the dendrite. And why is it good to have a positive signal? Well, if it's really positive, that means it'll travel a far distance. And hopefully that positive signal will get down to the spike initiation zone, and it will cause an action potential to fire. But we're not worried about that right now. We're still focused on this. So, that is an example of temporal summation. So what's another way to get a big positive signal? Well, you could have one positive signal going here, and then you could have another positive signal going here, and you could have another positive signal going here. And then what happens is when this one, and this one, and this one all reach the same point, they too are going to add together into a very positive signal. So in temporal summation, what's happening is you're getting a lot of positive signals that are added together because they're occurring within the same time. In spatial summation, what you're getting is a lot of positive signals that are adding together because they're occurring close to one another in space. And so they add to one another, and then that makes a very big signal, and again, hopefully that'll propagate down here and cause an action potential. But this is spatial summation. Okay? So, just to make it simple, to offer one final drawing to this effect, if we had a candidate axon here, and we had a bunch of signals that were firing, high frequency, that added up into a very big signal here on the axon, that would be temporal. Whereas if we had instead a few signals that were firing here, and then a few signals that were firing here, and then a few signals that were firing here, that all added together at about here and created a very large excitatory signal, that would be an example of spatial summation. So spatial summation is adding them together because they're close in space. Temporal summation is adding them together because they occur close in time. Alright. Last question I received is at 7:30, so 7:34, so at the very least we'll be going up until 8. And we usually try and take a break around 7:55 just so I can catch my breath, but so we'll be going at least until 8. Alright. Next question. Would it be correct to say that nicotinic receptors are activated by acetylcholine in open ion channels, while muscarinic receptors are also activated by acetylcholine, but are coupled with G protein? Yeah. That is correct. That's pretty much all there is to it. I'll illustrate this just for the sake of everybody's benefit, but that's correct. So, if you have a nicotinic receptor is an ion channel, and a muscarinic receptor is, well, it's a metabotropic receptor, which is a fancy way of saying it's sort of like a G protein coupled receptor. Again, for the purposes of this exam, you can assume that they function in a roughly similar way. So, if you had your ion channel, your nicotinic acetylcholine receptor, and you had your muscarinic acetylcholine receptor, remember that each of these can bind acetylcholine. So that can bind to the nicotinic receptor, or it can bind to the muscarinic receptor. Likewise, you can have things like nicotine bind to the nicotinic receptor, and you can have muscarine bind to the muscarinic receptor, but acetylcholine can activate either of these. And when acetylcholine activates the nicotinic receptor, acetylcholine receptor, you're going to get free flow of ions in and out of the cell. You guys are probably sick of seeing this by now, but so it goes. So you can get free flow of ions. That's the nicotinic acetylcholine receptor. With the muscarinic acetylcholine receptor, when that's activated, it'll activate different effector proteins, second messenger cascades, and cause all sorts of great stuff to happen, including but not limited to, well, I don't want to confuse you guys, but it'll cause widespread changes in the efficacy of other ion channels, and also it'll sort of work as a means of potentiating the entire cell for being more receptive to subsequent stimulation. So the effects of that muscarinic receptor are much more broad than the effects of activating a simple ion channel receptor such as the nicotinic acetylcholine receptor. The only thing I would be wary of in the wording of this question is that you said here, say that nicotinic receptors are activated by acetylcholine in open ion channels. Nicotinic receptors are ion channels, so they do open ion channels because they open themselves. They don't. A nicotinic receptor does not go down here and then activate another ion channel. If anything, a muscarinic receptor might do that, but a nicotinic receptor won't. So a nicotinic receptor will just activate itself and allow for the free flow of ions. Yeah. I'm aware of the fact that there are advertisements that pop up, and occasionally they will pop up right as I get to your question. A way to deal with that is just download Google Chrome and then download an extension called AdBlock. If you put either of those into Google, it will direct you right to that. But unfortunately, I fear that that's a necessary evil unless I want to spend a couple $1,000s making this a professional Ustream channel, which perhaps I shall do in the future. But do not forget that, as you said, you can always watch the YouTube videos, assuming that technology cooperates with us. Alright. Question. Can you clarify the different divisions of the lateral pathways of the spinal cord, and what type of information is conveyed in both the lateral pathways and the ventromedial pathways? What is the difference between these two pathways? We did not get to cover the descending motor system in as much detail as maybe we could have. So there's not going to be an incredible amount of information on the exam about the descending motor system. I'm just going to pull up an image here of the lecture that was relevant to this. Where is it? Okay. So what this question is basically talking about is this slide here. So different divisions of the lateral pathways of the spinal cord. There is the cortical spinal tract, and then there's also the rubrospinal tract. I'm going to see if I can make this full screen, and I hope it doesn't destroy the Ustream. I'm just standing by for a couple seconds to make sure that that pops up okay. Okay, good. So different divisions of the lateral pathways of the spinal cord. So there is the cortical spinal tract and there's the rubrospinal tract. Actually, the easier way to answer your question is to answer the second part first. What type of information is conveyed in both the lateral pathways and the ventromedial pathways? So all of that is motor information. The lateral pathways control of voluntary motor movements. Just going to go ahead and actually get the pin here. So the lateral pathways control of voluntary movements of distal musculature that are under direct control. So motor here, and then the ventromedial pathway involved in control of posture, locomotion, under control of the brainstem. So again, motor. So for both of these pathways, lateral and ventromedial, both of these control motor movements. Or in the very least, they convey motor information. The keyword is motor. Once again, motor. So lateral pathways and ventromedial pathways, motor. That's what you have to know about them. Now, in the lateral pathways, there are these two different divisions here. There's the cortical spinal and the rubrospinal tract. And the cortical spinal tract is involved in voluntary motor movement, and the rubrospinal tract is involved in sort of the control of movement. Interesting, the rubrospinal tract is a very ancient, very primitive tract that goes up and down. I think you can see it in like some really old lizards and stuff like that. But don't worry about that. Again, here, movement, motor, movements. So all of these are motor pathways. And we may, if we have time, we may go back in and try and cover these again and cover a little bit more information with regards to, certain diseases and how you can have disruptions of these different pathways going down. But that's all I would like you to know for this exam. Descending motor pathways, lateral, ventromedial. Very good. What's different between the two pathways? Well, as I said, the ventromedial pathway controls posture and locomotion, and the lateral pathway controls voluntary movements, distal musculature, that are under direct cortical control. So that would be like your hand, or more like your wrist, I would say. Don't know why I felt compelled to draw something there. I'm pretty sure everybody knows what a wrist is. Right. Okay. Is it correct that the spike initiation zone is simply the point at which the action potential is initiated? Yes. Yes, that is correct. I don't really think we need to complicate it more than that. The spike initiation zone is the point at which the action potential is initiated. What is a quality of the spike initiation zone? It has a high density of voltage-gated sodium channels, and those voltage-gated sodium channels respond to the change in membrane voltage, and they cause the massive influx of sodium, the depolarization that begins the action potential. Alright, I do not have any more emails right now. So what I'm going to do is I'm going to take a 10-minute break and turn off the stream, save the video file, and I'll just wait for some more responses. And if come 8, I'll start up the stream again. Oh, I just got another email, so we have something to do when we come back. But I'll start the stream again at 8 o'clock, and then I'll answer any of the questions that we have. And if I get any more questions for them, we'll just keep on going, and I will see you all at 8.